Hello, let's start maybe. And welcome to lecture 6 of SI2720. So you've had lectures so far, I think, by Eric Lindahl only, but we're 3 teachers in this course. So it's also me. I'm Lucy Delamat. And you will see Berg Hess, I think, later this week. And then I'll come back and Eric will come back and so on. In principle, we know what every person is covering. But today I'm going to build onto what Eric has been teaching you about and cover membranes and membrane proteins. I will say I'm attempting to record this lecture, but last year I failed twice to record it. So it's possible it won't work. For this lecture, there's the lecture from last year, so worst case you have this, and it hasn't changed too much. But yeah. So this is a really nice system, but it's almost as stressful to get it to work than actually kind of giving the right lecture. Okay, so just to give you a little bit of context, this is probably, I guess most of you are physicists in the first place, and you're taking this class on biophysics. So this will be maybe the most biological class that you've had up until now, which is cool because you're going to discover many new things, but it's a lot of vocabulary and so on. So I hope you feel comfortable interrupting or asking questions if you need clarifications, if I'm using words that you don't understand and so on. Okay, so I'm comfortable being interrupted. And I'm not going to go through study questions from the previous classes, but there will be a few notions that kind of come up in this lecture that you've covered before. So I might ask you a few questions about different things that you've studied, and then we'll see how well you're doing with the previous concepts. Yes? Okay, so today is membranes and membrane proteins. We're going to cover, in principle, 4 things. And let's see how much we have time to cover the 4th point. I mostly want to just give you some overview. And so we will be talking about how the membrane is structured and how that supports its function. We'll be talking about membrane proteins and how they are structured and how they function. And then we'll talk specifically about the basis for membrane transport and signal transduction, which are 2 aspects of what membrane proteins do in life and specifically in the human body. But I guess we'll talk a little bit about plants and other living systems too. It builds quite a bit on what you've learned about amino acid properties, about secondary and tertiary structure, about structure-function relationships that you have started covering in globular proteins. You remember globular proteins were proteins that are mainly found in solution in water. So they're quite a little bit different from membrane proteins. We'll see how and why. But they will be built from the same kind of components, both in terms of amino acids and secondary and tertiary structure. So this will come back. But we start with the membrane. So even in the absence of these membrane proteins, we'll talk first about the membrane. So you might imagine a cell to function has to be isolated. That means that whatever happens inside of the cell cannot communicate too much with the outside. It had to be contained. But then the cell still is not working in isolation, so it needs to be able to communicate information with the outside. And so this will be what we're discussing a lot today. So you might imagine that something that contains the inside of the cell should be very robust, kind of like a house that isolates the inside of the house from the outside of the house. And so it's actually very surprising that the structure that isolates the inside of the cell from the outside of the cell is a very fine structure. And in fact, we didn't know for a very, very long time that there was such a structure that isolated the inside of the cell from the outside of the cell. And this is because the cell membrane is so thin that you couldn't see it with light microscopy because it is smaller than the diffraction limit. So the size of the membrane will be of the order of 5 nanometers or something like this, which is smaller than the diffraction limit for light, right? So you can see it in a regular microscope. But then there were progress made in electron microscopy. And these are some of the first images that were found through electron microscopy after staining the tissues, I think specifically with heavy metal osmium. And what appeared is something quite surprising, is that all the membranes, whether you were recording from different types of cells, they showed this kind of fine structure where you could see kind of 2 layers. And you can see the same thing on here. So basically, this heavy metal was localizing to 2 kind of things that we don't know what they are for the moment, and not in the middle. And it's actually also interesting that this is the membrane of a cell, delineating the inside from the outside. But inside the cell here, you have another kind of membrane-like structure, because it looks very similar to what you can see here. And so at that point, I guess the scientists realized that there were membranes that were not only separating the inside of the cell from the outside, but also inside of the cell there were additional membranes. And apparently they had a similar kind of structure. So do you guys know, have you ever heard what this is? What it represents? No idea. Good. Then we'll go through it. But before we go through what these 2 layers are, just keep in mind that this is important for their structure. I just want to give a few ideas of what the roles are, and I already discussed one, which is isolating the inside of the cell from the outside of the cell. So this is called compartmentalization, which is a difficult word to pronounce. And basically what it is that it basically isolates the inside of the cell from the outside. Then the membrane is also a scaffold for biochemical activity. This is what you can see on this picture. And the idea for this is that within a cell, right, there's a lot of activity that has to happen. Maybe you're familiar with enzymes or things like this, which means that the proteins or the nucleic acids, they have to find one another to function. And if you think about the cell as a 3-dimensional object, you can imagine it's very difficult for like a protein to find its partner, because there's a lot of degrees of freedom. But when you put in a 3-dimensional object, a 2-dimensional surface, then it reduces the number of degrees of freedom where the objects can find one another. It's just a geometric argument, right? So it's more efficient, basically, for the cells, for the objects to find one another. So this would be what is called scaffold for biochemical activity. Then a third thing that is important and is actually related to number 4 is what I was mentioning very early on, is that the membrane has to isolate the cell inside from the outside, but also it has to enable the cell to communicate with the outside, because it has to receive signals from the outside, and it doesn't work as an isolated object. So role 3 and 4 are providing a selectively permeable membrane. For example, this is a plant cell. It has to be able to import water. It's important for its function, but it might not want to let in other things than water. That's called selectivity permeable. And then the cell also has to be able to transport solutes. So here in this case, it's a proton, but we'll talk later a lot about other types of ions like sodium and potassium, or glucose, or other things that are important for biology. And then the membrane has to let the transports go in and out, and sometimes it has to provide energy to transport them. We'll come back to that quite a bit. It responds to external signal, as you can see here. For example, is highlighted a hormone would interact with the cell membrane and trigger something inside the cell that would trigger a response. This is the 4th part of the lecture. And then 2 other things which we're not going to talk too much about is intracellular interaction. So cells might be directly linked to one another, and then it's the membrane that forms the interaction directly. And also it's involved in energy transduction. So like I said, today we're covering compartmentalization and these 3 topics. And we've already started. So this is the answer to this question I was asking you. Do you know what this membrane is like? Why do we have 2 layers? In kind of, long time ago, Overton was one of the people who realized that it must be that the outside of the cell is hydrophobic because he measured basically how different types of solutes were entering plant root hairs, and he was basically able to build a correlation between the lipid solubility of the solutes and how fast they enter the roots. And so then he came up with the idea that it must mean that the surface is hydrophobic. He was right. But this was very kind of preliminary. Then a little bit later, beginning of the 20th century, this was a very cool experiment. So what these guys did, they're from the Netherlands, they crushed together red blood cells, and then they measured on a device like this how much of the surface of the cell was contained compared to what they measured as the surface of a red blood cell. So if you look at this kind of device, what it is it's like a square that contains water, just a solution, and then they put the extract of these cells on top of it. And basically what it looks like is like this. And the reason it looks like this is that they found out through these experiments the membranes, they're made of lipid molecules. And lipid molecules are kind of really interesting because they're made of two parts. They're made of a hydrophobic part, the hydrophobic tails, and a hydrophilic head group. So the hydrophilic head group tends to arrange itself towards the water. That's thermodynamically favorable, right? And the hydrophobic part tends to organize itself away from the water, so towards the air, in that kind of arrangement. And so this device contained a piece of wood that you could displace so that you could measure basically the area that was covered by this kind of thing. And what they found, which was really surprising at that point, was that the surface of water that was covered was equal to twice the surface of the cell. And with this, they were able to tell that the membrane is made of two layers of lipids arranged with their tails looking at one another. Yes?
Some organelles have double membranes. Is it the difference that there's an extra?
Oh, you mean like mitochondria, for example, like this one? Yeah, exactly. So yeah, I'll answer this in a second. So in the plasma, let's go back to this actually. In the plasma membrane, right now you see these two black lines are where the heavy atom like to bind, so it's the head groups, and the tails are white. And so then the head groups, which are again hydrophilic, they're arranged towards the solution, and the hydrophobic part is just arranged the two lipid molecules coming together. So there's nothing that's unfavorable thermodynamically because the hydrophobic parts come together. They're not exposed to water. In the intracellular membranes, it's the same kind of story. And I think you're referring to mitochondria, for example, which we're not really going to cover today. But then in that case, you basically have two membranes. And the role is to isolate let's say the purple region from the brown region. But each membrane, here and here, is made of the same arrangement. So basically it kind of looks like this if you want, right? Mm-hmm. So good question, you do have two membranes. So this is a view of what it might look like extracted from anatomistic simulations. You have the head groups the tails in green, the head groups in red, and the solution in blue. So you can really clearly see that this is a way such that the tails that are hydrophobic, they don't come in contact with the hydrophilic water. Is that clear? Yes? Because basically there's two equilibria. If you have water on the other side, then you're able to make the surface that is half the size, right? But if you don't have water, then they will start arranging them in an unfavorable type of environment. So the only place where it's thermodynamically stable is when you have a surface that is made of a monolayer like this. So this adjusts directly. Okay. So this is the really basic idea, because the main component of these membranes are the lipids. But throughout history and many, adding layers of experiments, mainly actually due to measurements of surface tension and comparing what was the surface tension of a pure lipid bilayer and biological membranes. Several researchers a long time came up with the idea that there must be other things than lipids in the membranes, and specifically the hypothesis was that membranes must contain proteins. And in those days, 1935 to 1950s, it was really not known what proteins really looked like. And I like this picture because it looks, when it looks a little bit ridiculous, but it was kind of a creative idea. So the idea was that you would have lipid molecules and then proteins that were kind of coating the lipids, and that would make pores. Because we said before, right, that cells need to be able to transport water and so on. It turns out this picture is wrong, but it was, a good idea for when you were in the 1950s. But the truth is not that far, actually, from it. So the truth, and this is a model that dates back from 1972, we still think of the membrane in the same way. So in this model, basically, you have proteins that are arranged. You remember these globular proteins? So they're arranged in helices sheets that come together as globules. And they are arranged within the membrane. And we'll come back in a lot of details, actually, to what they look like and why. So this would be a more colorful picture where you have the lipids with their head groups, their tails, bilayer, you can recognize it, and different types of proteins that are embedded inside. Remember, this is about 5 nanometers, 50 angstrom, so it's extremely thin. And most of the proteins kind of are of similar size to that. What you can see from this picture, and that is quite crucial, but you can maybe tell from the name, fluid mosaic, means that this is not a static assembly. So all these molecules, they're arranged in a way that is thermodynamically favorable, but they're not linked covalently, right? So they can move around. So each of these lipids is kind of moving around, diffusing, and there are really nice experiments that can show this. The proteins also move. And when we come to the last part of the lecture, we'll talk about signal transduction. You'll see that proteins need to come close together and separate and so on. So this is a dynamic assembly, which is difficult to see in a picture. But now we have nice molecular dynamic simulations, actually, where you can see this also kind of clearly on the atomistic level. Are we good? OK. So I know you guys are not chemists, so I'm not going to rub this in. But I will say a few things about the chemical structure of membrane lipids, just to give you an idea of the variability. So this picture that I already showed you holds all the time. But then all the cells are different, meaning a heart cell is different from a plant cell is different from a bacterium. And also the membranes from the plasma membrane, so the membrane on the outside of the cell and the inner membranes are also different, which means that the lipids can be quite different. So they will always have a hydrophobic part and a hydrophilic part and a linkage in the middle. But then the chemistry can be quite different from one to the other. So the tails are often the same. So there's often, actually, I think almost always, 2 per molecule that are really long tails. You can see better here the ratio, actually, of the size, right? So it's very long hydrophobic tails. And then the head groups can be quite different. So this is a list of phosphatidyl phosphoglycerols. And they are called that because they have a phosphate group that makes a link between the chains and the head groups. And the head groups can then be either very small with just the phosphate, nothing else, or you can have different types of chemical groups. And so they're slightly different, but they all have the same property, that they're hydrophilic, right? Okay, so just similar in a way and different enough so that you can get different biological function. Yes?
The lipid tails, are they saturated or unsaturated?
So often in biological media, but it's not always true, there's one chain that is unsaturated and one chain that is saturated. For those of you who are maybe less familiar, saturated versus unsaturated means that the bonds that form the links between the carbons can be either single or double bonds, and then it gives different properties. You might have heard about all these polyunsaturated fatty acids. You should eat salmon because it's good for your brain, like that sort of thing. This is usually high unsaturation contents. So there are specific lipids that have a lot of unsaturation, and they're supposed to be good for memory formation. I think it's true, and I think it's not very well understood why, but yeah. Then this class here are called sphingolipids, and they're similar again because they have a hydrophobic part and a hydrophilic part, but also slightly different. And these ones, for example, contain sugars, which is a different kind of chemical object, and they're important for different types of biological functions, including actually very important for bacteria. And then one extra component that we haven't talked about is cholesterol. You've heard of cholesterol, that it's bad for you or it's good for you, depends what kind. In the context of lipid bilayers, these cholesterol molecules, they're represented in green, whereas the lipids are shown in gray with the head group in kind of pink and red and orange. You can see that they kind of bury inside the tails, and they will typically make the membrane a lot more rigid. And so this is the opposite from the polyunsaturated fatty acids. They're more rigid, so they make the membranes more rigid. Sorry, they're not more rigid themselves, but they make the membranes more rigid, which is typically bad for brain function or clotting and so on. So this again is complex, so you can't say because you have high cholesterol you'll get this kind of disease, but in general cholesterol has the tendency to rigidify membranes and so to make them less plastic. Okay. Good, so this concludes what I wanted to tell you about just the membrane itself, and now I'm going to go into membrane protein and specifically their structures. So you've seen in that fluid mosaic model that we had the lipids arranged in this bilayer fashion, and then we had a lot of proteins, and I told you everything is fluid. Now just introducing some vocabulary, we have basically three types of membrane proteins. We have so-called integral proteins, peripheral, and lipid anchored, and basically this is what they look like. So the integral proteins are also called transmembrane, and basically what they look like is that they so-called span the membrane. So they have, they're mostly in a transmembrane position, and they have some domains that are outside and some domains that are inside, but they are, the bulk of it is made of the transmembrane parts. And they're usually alpha helical. We'll come back to why that is. Peripheral membrane proteins are proteins that are associated with the membrane, but they are either on the outside or on the inside, but kind of stuck to the membrane through non-covalent interactions. And lipid-anchored proteins are basically covalently attached to the membrane. So this is, for example, GPI-anchored protein. You can see the protein would be shown by this, what we call this, like ball of yarn. And it's attached to a lipid through a phosphoinositide linker. And you can see that then this protein will not be able to detach from the membrane because it's attached basically to a lipid, which is an integral part of the membrane. For most of the rest of the lecture, we're going to be talking about these integral membrane proteins, and the reason is that those and those, they look a lot more like globular proteins because they're not embedded in the membrane. Okay, so related to the function, I was telling you before that the the membrane has to isolate mostly whatever happens in the cell from what happens outside of the cell. And I've told you that this is a dynamic assembly, right? Now, if you think about it, we don't want spontaneously chemicals to go through the membrane, which means that the membrane lipids and the protein, they have to be very tightly associated, right? They have to touch in a way. Because otherwise, if they're not touching, you would have vacuum, water, I don't know, and then things would be able to go through. Yes? And actually, this is something that we are looking very closely at in research, is that there's new evidence kind of emerging in the last maybe 5 to 10 years that lipids, they're not only kind of just a place where membrane proteins localize. They are even, they take part in the membrane protein function, and they can regulate membrane protein function. So I don't have time to talk about this. You need probably a class on membrane proteins to kind of reach that point. But it's kind of interesting that biologists came from the idea that, you know, you had lipids and proteins, but reality, as always, I guess, is that things are more complicated and that, you know, different objects work together to build a complicated picture. What you guys need to know is that there should be a tight association to prevent things from going in between the lipids and the membrane. So this takes us back a little bit to what you guys have been learning in the previous lectures. We're taking an example of a protein called glycophorin A. And this protein looks like one transmembrane helix, an intracellular domain, and an extracellular domain. So this is an integral membrane protein or a transmembrane protein, right? Because it kind of goes through the membrane. And like I said, it's a single helix. And here you have the different residues. And you can see, if you have memorized the different amino acids, that all the ones represented in red here are hydrophobic. So this says isoleucine, leucine, valine, alanine, methionine, phenylalanine. So all of those are hydrophobic residues. And of course, that makes sense, right? Because these are the lipid chains. The lipid chains are hydrophobic. So in transmembrane position, you should have hydrophobic residues. On the other hand, completely opposite to this, the residues that are here associated with the head groups, they're not hydrophobic, let's say. So here you have 1, 2 lysines, 1, 2 arginines. Remember, lysines and arginines are at pH 7 positively charged, which means that they are favorably interacting with the head groups, which are hydrophilic. Does that sound familiar? No, as far as I can tell. Okay, but then still I can see here that there are a few residues that are not in red. So there are 3 here. Glycines, you guys remember what glycines are like? Say again? Yeah, so the side chain, it's the smallest side chain. It's only 1 hydrogen in the side chain. So it's very small, and it's very flexible, and it's symmetric. And I think you heard in one of Eric's lecture that the Ramachandran angles that it can take are much wider because it can be very flexible, and there's no constraint, so it can basically rotate in all dimensions. And I'll come back to that, but maybe it's not an accident that you have so many glycines here in the middle. Because glycines are so flexible, which means that, you know, the helix is not as constrained as it could be, so it can be a little bit flexible in the middle. And if this protein needs to come close to other proteins, then these glycines are going to help it accommodate the rest of the proteins. And then you have 3 other residues here that are in blue, threonine and serine, which I guess you've gone through, but maybe you don't remember, and that's okay. These are polar. They have a hydroxyl group, OH. And I've just told you, in principle, it should really not be favorable to have polar residues in the hydrophobic region, right? And of course, most of them actually localize close to the head group, so that is more favorable. But we'll see in a second that it's possible actually to have a few hydrophilic residues in the transmembrane position. It's all a question of thermodynamic balance, but mostly you have hydrophobic residues in the transmembrane region. Okay? So what did you learn about the globular proteins? I guess you learned that you can get helices that assemble together. You could have beta sheets where you had these folds where you have several helices and then sheets on the side. And every time you were isolating the hydrophobic residues from inside of the protein and putting the hydrophilic residues on the outside, right? Because the protein needed to be in solution. See how this is different? We're not trying to isolate the hydrophobic residues from the environment. Instead, we're exposing them to the hydrophobic environment because the environment is different. But the thermodynamic argument is really simple, right? It's just that things that dislike water tend to stick together, and things that tend to like water just stick together. And related to this, I don't know how much you've talked yet about determining protein structures. Eric is a big fan of cryo-electron microscopy, so it's possible that he mentioned it several times. There's also X-ray crystallography. And membrane proteins have a challenge when you try to get to their structure and do this kind of structure determination, is that imagine to be able to get their structure, you need to extract them from the membrane, right? You need to get high concentrations. And if you just take this protein and strip it away from the membrane, what do you think will happen? It will lose its conformation. Why?
Because it's the hydrophobicity.
Exactly. That's right. So if you take this protein, now we can imagine that all of this was hydrophobic because it was exposed to the chains. If you remove it from the lipid, it's going to collapse onto itself to isolate the hydrophobic residues from the water. So then you're not going to have the structure that you think you're looking for. You're going to have a distorted structure. And so what people have come up with, and people actually got the Nobel Prize for this, was to solubilize this into a detergent. And what is a detergent? It's basically soap. Soap has a hydrophobic tail, a hydrophilic head. And so basically the hydrophobic tails will come and stick around the hydrophobic part of the protein and orient the hydrophilic heads towards the solution to keep the protein in its original configuration. So this was really key to understand how membrane proteins are structured in their native environment. I don't know if I actually motivated this very well, why it's so important to understand membrane protein function. The key thing really that everybody cares about is that they're really good drug targets because they're on the outside of the cell, right? So it's easy to reach them and to change the way they function. So there was a huge competition to understand what they are structured like in the context of developing drugs for them. But we'll come to GPCRs soon. Okay, so I'll finish this part before we take a break. This goes back to our glycophorin A. Remember, this was our protein that had an intracellular domain and then a transmembrane part and extracellular domain. On this graph, you would have the sequence here from the N terminus to the C terminus, so all the amino acids just arranged according to their number. And on this axis, you have the hydrophobicity of a short stretch of amino acids centered around the residue. So for example, for residue 50 here, you have a hydrophobicity of minus I don't know how much. And that corresponds to the hydrophobicity of the stretch that is around amino acid 50. And you kind of do this for all the amino acids. And you can see that the region that has hydrophobicity that is high would be the transmembrane part. This is amazing, actually. If you just take any sequence, put it just in a server that calculates this hydropathy, you can predict very, very nicely what are transmembrane regions. So without knowing the structure of a protein, you can often really say this is a protein that contains 3 transmembrane helices. It works really very well. And then there's another rule that is about the positive inside rule that was discovered by our colleague at Stockholm University. And it says that due to the way membrane proteins fold, the most positively charged amino acids are located in the intracellular domain. And this is what we could see on this picture, right? That you had all these positive charges that were localizing inside the cell. And like I was saying before, it is possible to find a few amino acids in transmembrane position. It's not favorable, but if you have enough hydrophobic residues around it, then it's possible to bury hydrophilic residues because the overall hydrophobicity of the stretch is still favorable to put in transmembrane region. Plus, sometimes you also find helices that come together, and then in that case, it's possible to bury the hydrophilic residues inside of the assembly of the protein itself. A little bit like in the inverse of the globular protein, where you are burying the hydrophobic residues, and that way you can bury hydrophilic residues inside of the membrane. It gets a little twisted, but biology is just has to make things work somehow. I'll say basically nothing about this, just so you are aware. In bacteria, it's possible to find transmembrane proteins that are formed of beta sheets, anti-parallel. So here, this is one beta sheet that folds back into here. So you can see that they're arranged in a barrel shape. They're basically only found in mitochondria and chloroplasts, and they serve as channels to import and export hydrophilic compounds. And after the break, we're going to talk a lot more about this phenomenon of transporting things across the membrane.